Hey everyone, today we're going to talk about genetic engineering. So first, what is genetic engineering? It is the modification of DNA by adding or removing sequences of DNA. And through the use of genetic engineering, we are able to produce transgenic organisms like the ones on the right side of the screen here. These are animals that all contain foreign DNA, so DNA that is not originally found in their genome. We can also use genetic engineering to produce gene therapy, and we can also do it to clone genes as well. Now, back to these organisms here on the right side of the screen, these animals are all modified to contain a glow-in-the-dark gene found in fireflies. So you can see the pig's nose glows up, the rabbit glows in the dark, this cat's nose and ears are glowing, and these monkeys, their skin is glowing because they have all been, their DNA has been modified and injected with DNA from that firefly gene. So how do we genetically engineer DNA? So how do we change it? We use restriction enzymes. If you recall from yesterday when we talked about DNA fingerprinting, restriction enzymes are used to cleave open or cut open DNA. Now in this case, we're going to be exposing human DNA to restriction enzymes in order to isolate a desired gene. And for this example, we're going to talk about the human insulin gene. So we're going to use restriction enzymes and cut on either side of that DNA sequence in order to extract the full intact insulin gene. Now we're going to take the same restriction enzyme and this time apply it to a bacterial plasmid. And if you recall, a plasmid is that circular ring of DNA that's found in bacteria, and it's only found in bacteria. And the reason that we're using that plasmid is because we're going to use it as a vector or a means of transporting the desired gene, and in this case the human insulin gene, into our target organism. So to recap, we've cut open the plasmid and we have isolated the desired gene. Now that we have both of those things, we are going to combine them together. And so we're going to the two open sides of that plasmid, we're going to stick in the gene, and now we have something called recombinant DNA. That's DNA that has been recombined to contain foreign DNA. And this recombinant DNA plasmid is then reinserted back into the bacterial host where it will reproduce, right, every 20 minutes. And in the process of reproducing, it will also make whatever desired proteins this bacteria makes, plus the protein that we want it to make, the insulin. And then we can extract that insulin and give it to people who have diabetes. And that is actually how we get the insulin to treat people who are diabetic. So here's a visual of what I was just explaining to you. So up here, we have an animal cell. Okay, we're going to say this is a human cell. Okay, now over here on the right, we have a bacterial cell, right? And we can tell this little circle right here, this is the plasmid that I was talking about. So we've isolated our human cell and our bacterial cell, and you can see that this arrow comes down, and you can see this little itty bit, kind of looks like a little bit of a wishbone. It's not. It's actually the desired gene. In this case, we've extracted it from the DNA. And we have over here, you see the plasmid is no longer circular, it's now broken open because we've cleaved that plasmid. And now if you look, the plasmid comes down, the desired gene comes down, and they're added together to form recombinant DNA. And then we reinsert that plasmid into our bacterial host, where you can see that it's blowing up that gene that we wanted. Oh, I guess I identified this is the rest of the DNA in the bacterium. And then we have the desired gene. It's the same gene that was up here. Okay, we have that desired gene. Then we have a single strand of nucleic acids. That's the mRNA. So right here we're trying to model the process of protein synthesis. So we have our desired gene, it's converted into mRNA. Then translation happens, right? We've got our ribosome. And then lastly, the squiggly ball, that's going to be our protein. In this case, the insulin that we were looking for. So that's genetic engineering. Now, why is it possible for genetic engineering to work? Well, DNA is similar in all organisms. It's all double-stranded, and they have the same 4 nitrogen bases. That's the first thing. The second thing is that the same restriction enzyme was used on both samples to get the desired gene and also to open the vector, and in this case, the plasmid. So because of that, the complementary bases are unattached when we cut them out, right? You can see in the picture here that we have some of these nitrogen bases that are not paired on both ends. And you will notice that their complement is found over here on the desired gene. So when I insert this plasmid over here, or if I insert, the desired gene over here into the plasmid, they get stuck together because we have those complementary bases pairing up and sticking together to form, again, a fully intact plasmid, this time containing foreign DNA. I mentioned earlier a type of genetic engineering known as gene therapy. So we're going to talk a little bit more about gene therapy. It is a form of genetic engineering used to treat human genetic diseases. And there are 2 diseases in particular that you need to know how we can help. The first is cystic fibrosis, and the second is SCID. Cystic fibrosis, if you don't know, is a disease of lungs. And people who have cystic fibrosis have an abnormally large amount of mucus production in their lungs compared to the normal individual. And that mucus can lead to lung infections and poorer health compared to the average human. SCID stands for severe compromised immunodeficiency, and this is the scientific term for a person who does not have an immune system. So we use gene therapy to help treat those diseases. We first have to identify that a person has the defective gene, and then once they have, in fact, tested positive for that defective gene, they will then be given a normal functioning gene that gets inserted into their DNA to help produce the correct protein and reverse the effects and symptoms of their diseases. Now, to inject that person with the normal functioning gene, they're given the equivalent of a vaccine, but it's not a vaccine. It's a transgenic virus. So that virus is not going to make them sick. So if it was the flu virus, it's not going to make them sick with the flu, but it will inject them with the normal functioning gene by randomly inserting that gene into their genome in every single cell of their body. And by doing so, when it's activated, when that gene is activated in the lungs, it will help alleviate the symptoms of that mucus production in a person with cystic fibrosis. The downside to this and the complication is the fact that it is randomly inserted. We are not able to tell that virus specifically where to insert the normal functioning gene. So by randomly putting it in different cells, some of the cells are going to have genes disrupt other normal cellular processes and can cause other secondary problems like cancer. So people who have cystic fibrosis or SCID and get treated with gene therapy tend to have a higher risk of later developing another type of cancer like leukemia. So what are some applications of genetic engineering? Well, we just talked about one, gene therapy. Another one is the application of medicine, producing large quantities of human proteins cheaply. There are 2 main proteins that you need to know are used through genetic engineering to be produced and given to humans. The first is insulin, like we talked about in how to genetically engineer an organism, and also the human growth hormone. The reason that we do this, even if you know somebody who has diabetes and they get insulin, it's still a little bit expensive, but not nearly as expensive as if you had hooked up some person named Susie, gave her a bunch of sugar, and then extracted her insulin as she's producing it. That would make insulin extremely more expensive than it already is. But by using bacteria that are relatively cheap and abundant, and being able to genetically engineer them to produce these hormones, we're able to make them at a much lower cost and a lot faster than if we used another organism to produce these hormones. Another application would be in agriculture, producing plants that are genetically modified to have pesticide resistance, herbicide resistance, and higher nutritional value than they normally do. Because if I'm a farmer, I want to make sure that I can spray herbicides and get rid of all the weeds without killing my plants. I also want to make sure that I can kill off any insects that want to eat my food, again, without killing my plants. And if I'm growing something, let's say corn, that doesn't really have a nutritional value, I want to be able to insert genes into that food that would improve the nutritional value and make it better for people who maybe don't have the means of being able to get a fully nutritional meal. So we call these organisms genetically modified organisms, or GMOs. So if you've seen that around or on labels or boxes, that's what we're talking about. And some examples of plants that have been genetically modified. Corn has actually, there's a lot of corn that has been inserted with a gene from peanuts, because peanuts are highly nutritious. And another is golden rice. And here's a picture of it. This is normal white rice and golden rice, which has inserted a gene of beta carotene into it, which makes it more nutritional than just normal rice. Another application would be to genetically modify bacteria to clean up after humans. So cleaning mining waste, the water that comes out of the mines, sewage treatment plants. We use a lot of bacteria to actually eat our waste in order to clean it and then allow us to reuse the water. And we've been able to genetically modify bacteria to consume oil from those large oil spills that you might have seen on TV. Now, with genetic modification, of course, comes the concerns with genetic modification. So the first concern is that new organisms that have been genetically modified could trigger allergic reactions. For example, that peanut corn that's in existence, a person who has a peanut allergy now might have an allergic reaction to corn when they never did before. But they're reacting to the gene from the peanut, not the corn. So now you have to be hyper aware of genetically modified organisms if you have a peanut allergy or another type of allergy. Also, there's concern that these genetically modified organisms, if they are released or somehow escape, could outcompete native species or create other problems for humans than we didn't already have before. An example would be that oil digesting bacteria. What if a person who was helping clean up that oil spill got their clothes contaminated with that oil digesting bacteria, and now that bacteria gets into your car engine? Well, that doesn't work, okay? And what are the long-term effects of eating these foods that are genetically modified? Are there things we need to be concerned about? Those are all valid questions. Now check yourself. What is genetic engineering? What is a transgenic organism? What is the purpose of restriction enzymes in genetic engineering? And what is gene therapy?